listeners of the Bio Report, I want to tell you about a new member benefit from the California Technology Council. CTC has teamed with Reprivata to offer members 6 months of Reprivata's COT network service for free, which gives companies the power of a VPN at a fraction of the cost. A remote, flexible workforce is the new normal, but most corporate networks aren't built to accommodate work from home at scale. Reprivata's COT network offers an easily deployable, affordable, and scalable solution to securely enable remote workers and protect the corporate network. To learn more about this and other member benefits, go to californiatechnology.org/member benefits. I'm Daniel Levine, and this is the Bio Report. While the revolution in genomics has led to rapid improvements in the cost and speed of sequencing and created new insights into the genetic drivers of health and wellness, proteomics has lagged behind. Being able to capture a comprehensive view of the changing levels of proteins in an individual could play a significant role in bringing about an era of precision medicine. SomaLogic is providing a push in that direction with its SomaScan Discovery platform, which can read 5,000 protein measurements in the blood through a single assay. We spoke to Roy Smythe, CEO of SomaLogic, about the role proteins play in health and wellness, the way the company's technology works, and the opportunity for it to help advance the area of precision medicine. Roy, thanks for joining us.
Thanks for having me.
We're going to talk about proteomics, SomaLogic, and the ability to run a test of 5,000 proteins in the blood to gain new insights into health and disease. I want to talk about 2 specific areas that you're working in, but before we do that, perhaps we can begin with why someone would want to sample 5,000 proteins at once. We think of looking for a specific protein that's a biomarker for a disease, but what's the benefit of looking at this whole realm of proteins?
Sure. Well, and that's really the crux of what's different about what we do here at SomaLogic, on this developing diagnostic side, and that is that we've known for a long time that proteins would be the best information source for human biology and potentially, to predict things about conditions and disease. But the problem has been, measuring enough proteins at any one time to get what I would call a full body signal. If you think about the comparator here is, we believe that since we can measure all the genes in the human body, that doing polygenic risk assessment, looking at the expression of all those genes may give us some information about someone's risk for things. We can come back to that later. As it turns out, that approach is not as good as we would hoped it would be. And the reason it's not as good as we would hoped it would be is because genes aren't dynamic. They don't change over time. They don't change with age. They don't change with, your genome is the same at age 25 as it is at age 45 and 65. Your genome doesn't change when you're sick. Your genome doesn't change when you take drugs. But all of those things change with your proteome. But the problem has been, measuring enough proteins at any one time to get sort of a full body signal, rather than measuring one protein at a time and then trying to correlate that to some disease process. The second problem has been, well, gosh, even if we could measure thousands of proteins at a time, how would we be able to make any sense out of it? And so the thing that's just happened to occur, and that SomaLogic happens to be at the leading edge of, is we figured out a way to measure thousands of proteins at a time rather than hundreds, is what almost everybody else in the world does in sort of a clinical commercial context. And then we have been able to use machine learning to look at those patterns of protein expression of those thousands of proteins and correlate them to things that you really want to know about.
Well, what exactly does your SomaScan platform do, and how does it work?
Yeah, so to sort of back up into the technology itself, what our founder discovered 30 years ago is that you can actually take little pieces of nucleic acid, of DNA, in solution. So when they're in the body's liquid format, these little pieces of DNA will fold into 3D conformational shapes. And he knew, in his research group, 30 years ago, that RNA, which is very similar to DNA in cells, actually does this. It folds into conformational shapes and moves proteins around in cells and has important roles. So his question was, why can't we just make a library of millions of different little pieces of DNA and select the ones out that, by dint of their shape, would bind to the shape of a protein? People thought it was crazy. And so and these little pieces of DNA, they're short, they're not genes, they're just little random sequences of nucleic acid, are called aptamers. And so he was the co-founder of the aptamer science 30 years ago. And what we do is we've developed a library of thousands of these little pieces of DNA. We've modified them to make them bind to proteins even more specifically and reliably. And because of that, if you will, we sort of engineered thousands of synthetic antibodies that can recognize proteins, but they're not antibodies, they're little pieces of DNA. And in the assay that we do, we expose a body fluid to these thousands of aptamers. We call ours SomaMers because they've been modified to work even better. And so we just happen to have currently 5,000 different of these, different forms of these. By the end of this year, we'll have 7,500. Your proteome has about 20,000 canonical, when I say canonical, I mean basic protein structures. But again, until we came along, you could only measure a few hundred at a time. So we expose a body fluid to a solution full of these SomaMers. They bind the proteins. That we throw the unbound protein away and the unbound aptamers away, or the SomaMers away. We then are left with these little pieces of nucleic acid bound to proteins. We then throw the proteins away, and we measure the DNA there we use an array, where an array is where you've got a complementary sequence printed on a slide, and you can identify whether or not that sequence was there in your specimen, because when it binds to the slide, it lights up. You can also do this with something called next generation sequencing. But what we do is we turn protein measurement into DNA measurement, and that's sort of the magic. As far as the tests themselves that are derived from this, the magic there is actually what's best described as pattern recognition. And so the way to think about the way the tests work, so you give me your blood sample, we run it, we expose it to our SomaMers, we figure out what proteins are in that sample and how much. We can do both relative to one another. And so now we have this pattern of protein expression in your blood for thousands of proteins. We've run this assay several 100,000 times on human specimens, and what we've done is we've collected this set of machine learning models that correlate patterns of expression of these thousands of proteins to others that have conditions that have known risks or have known diseases or known risks for diseases. And the way to think about it, the way it works, is a lot like there's something called radiomics, where machine learning has seen millions of images, and when a radiologist sits down to look at an MRI scan, some of these programs can now suggest diagnoses, even if the radiologist can't see it, because it sees a pattern in the film that it's seen millions of times before that the radiologist can't see. What we're doing is something very similar. We have a pattern of protein expression. Some of the proteins that determine these predictive models don't even have any known biologic relationship to the disease, but they're reproducibly there with others. And it's this pattern that can tell us things that we've never been able to know before.
Is sensitivity an issue? Are you able to detect proteins in extremely low quantities?
Yeah, that's the thing. So one of the problems with using, so the two technologies that are currently used to measure proteins exclusive of us are antibodies, the use of antibodies. And people have heard a lot about antibodies recently in the context of COVID-19, obviously. There's sort of the body's protein recognition system. And then the other way to do it is using something called mass spec, where you basically uh, use a physics methodology to sort of understand, what you're looking at in the mass spec machine uh, based on the physical characteristics of the protein itself. So the problem with antibodies is they can only measure um, currently in most contexts, hundreds of proteins at a time. It may be possible to measure a couple of thousand in some contexts, but it's very difficult. And antibodies cannot find super low concentration proteins. Mass spec can do most of this, but the problem with mass spec is that it's cumbersome, expensive, and not scalable or all that reproducible in regards to the results you get. So it is important. Once you get past the first 3 or 4,000 proteins, you begin to get into the proteins between that and the 20,000th that are difficult to find in part because they're low concentration. But as it turns out, some of those are important in these pattern recognition machine learning models.
We've heard a lot about the advances in genomics, kind of a Moore's law. Have we seen the same types of advances with proteomics?
No, proteomics has been a bit more of a slog, to be honest. And it's really interesting if you think about it. As it turns out, the genome is a fairly simple, chain of molecules, right? 4 base pairs, reproducible. We always know where it is, right? I mean, you can always find it in the nucleus of a cell. And so, as it turns out, measuring genes and then, finding ways to measure genes more quickly has progressed along at a very rapid pace over the last 20 years. As you said, Moore's law for biology has really been applied to that. Proteomics has been hard. It's taken us 20 years to get at SomaLogic to 7,200 or so by the end of this year of the canonical 20,000 protein structures. And the reason for that is that proteins, all 20,000 have a different shape. All 20,000 have a different half-life. They exist in different parts of the body. As you mentioned, they exist in different concentrations. And so the measurement of proteins, of thousands of proteins, has turned out to be a technically very challenging project. Has not moved along at a Moore's law pace, for those reasons.
Is the ultimate promise of this that you can, in essence, run multiple tests at once rather than looking for a specific disease presence?
Yeah, so there's several things about this type of test that are really fascinating. And my background is originally as a clinician and a translational scientist. And so when I got the call from the recruiter to look at this job a little over 2 years ago, I literally tried to take it over the phone with the recruiter, and she said I had to actually interview. But the promise for these tests is really amazing, and let's just walk through why that's true. The first thing to realize is that basically all of diagnostic medicine, if you think about it, so if you look at a CT scan, you're looking at proteins. I mean, you're looking at an image, a cross-sectional image of the body. Now, there's some minerals in there too because of the bones, but everything else is basically made out of protein, right? And so when you're interpreting a CT scan, you're indirectly looking at proteins in the body. When you take a blood pressure measurement, with a blood pressure cuff on someone's arm, you're indirectly measuring the action of proteins in the body, hormones, catalysts, reagents that are having an impact on. Some want to lower the blood pressure, some want to raise the blood pressure, some want to tighten up the vasculature, some want to relax the vasculature. You're measuring all of these protein interactions indirectly. When a doctor does a physical exam, when he looks in your throat or palpates your neck, he or she is examining proteins indirectly. And so what we've done here is we've just gotten right to the source. And because of that, some fascinating things seem to be involved. The first is that we're able to see risks that traditional diagnostics can't. So we know that, for example, in cardiac disease, 15% or so of acute events that people have, once they get up into their 60s or older, are in individuals that weren't thought to have any risk factors. We also know, if you're a doctor, treating patients, that even patients that you feel like you're treating at the top of your license, you're really leaning in on this patient. You're managing everything. It's a diabetic, you've got their sugar under control, they've got a little hypertension, you've got that under control. They're coming in to see you every 2 months. They're fit. We know that in that population, things still happen, right? It's just accepted in medicine that happens. And what we're being able to do with these tests is to be able to tell a clinician, this person may not look like they have any risk, but they do. And this person, you may think you're maximally managing them, but they still have risk. And then, of course, the ability to replace what I would call big box, more invasive, more expensive, less convenient diagnostics in some case is also an incredible, advance for both, safety, convenience, and mainly for cost of healthcare. So we have a test, for example, that can tell you if you've got fatty liver, something called NASH, and can even tell you what the histologic subtypes of that disease are. And the gold standard for that diagnosis is sticking a needle in someone's liver. We can tell you what your primary cardiovascular risk is for a heart attack or stroke over the next 3 years with a blood test without you having to get a non-exercise stress test, without having to have 3 or 4 visits to the physician. We can tell you what your VO2 max is, so what your aerobic fitness is, a thing that athletes and armchair athletes do sort of on an annual basis, without you having to get on a treadmill and exercise yourself to exhaustion. So the ability to see risk that's not discernible from other methods, and then the ability to replace diagnostics that are more invasive, more expensive, or less convenient is going to be transformational.
Well, what's the result that's delivered to a clinician? How much interpretation is provided?
Sure. So currently we've released, our first 10 of these tests. We have more than 100 in the pipeline here. These protein pattern recognition tests that can tell you things about either a current condition, like some aspect of your body morphology, like what's my visceral fat, what's my aerobic fitness, what's my total body fat. We can do all those things, and then also these disease predictions. So things like your primary cardiovascular risk and so forth. So we released the first 10 tests into the test market, what we call our test market, the concierge practices in the local Colorado area here right before the pandemic. And what we provide physicians and patients with is it depends on the test, of course. So in the case of total body fat, we give them a readout of percentages, and so your total body fat is in this percentage range, and they're relatively narrow ranges. and then for cardiovascular risk, we basically say you're either at low, medium, intermediate, or high risk. And then we quantify that with a clinician and with the patient, saying, it's a 1 in 100, 1 in 50, or 1 in 4. And so, we don't give them the list of proteins back. We do all that bioinformatics work here. We just give them a result back. We either tell them, for example, it's one of these health and wellness tests about body composition. We'll tell you what your percent body fat is. If it's a test for risk, we tell you, we bend that risk into categories and tell you, with something that you can understand just how high that risk is.
What have you done to validate the test?
So the tests, as it turns out, are heavily validated. We don't release them until we do a couple of things. So the first thing we do is we decide at the beginning of the development of one of these tests what the existing truth standard for diagnosis already is and how accurate that is, right? So what's the sensitivity and specificity of the existing diagnostic method? So, for example, if we're going to try to create a test that is as good as an ultrasound or a biopsy for liver fat, we then have to try to create a model that has similar sensitivity and specificity. And so what we start out with is we find out what the truth standard, the existing diagnostic truth standard is. In our pre-test development documentation includes, we're not going to release this test until it reaches this level of sensitivity and specificity. So that's the first thing we do. The second thing we do is that once we create a test, what we think has that sensitivity and specificity, and so we create these from, what we do is we take existing blood samples from patients that have these conditions or these risks or these body types. And so we'll develop the model from a known set of samples. And then what we do is we actually take an unknown set of samples, and we've got access to these because we've made arrangements with biobanks and pharma around the world over the last several years, to have access to these samples. We then take a blinded set of samples. So this is basically a retrospective virtual clinical trial, right? These are humans. It's no different from walking downtown Los Angeles and drawing 1,000 tubes of blood. It's the exact same thing. So we take a blinded set of samples and we run the test on those samples. And then we look to see what was the risk in that population for this disease, or what were the incidence of this disease. And only at that point, when we've validated that the model actually works as we thought it would with the appropriate sensitivity and specificity, do we release these.
Are these tests reimbursable?
So they're not reimbursable currently, through third party. So, this is a new diagnostic platform. And so currently, we can be paid for them under something called an LDT, Laboratory Developed Test CLIA license. And so we do have a laboratory certification that allows us, if a physician agrees that he or she thinks the test is valid, we can be paid, but we have to be paid. We're not paid by insurance currently. That takes a longer period of time, and we're walking down that path. One of the problems with achieving third-party reimbursement for diagnostics is that it's expensive and it takes a long time. And so, and the thing that's different about our platform compared to most diagnostics is that we're not going to market with a single test. Our company isn't built around a test that tells you if you've got colon cancer or not. We're going to have 100 of these tests on our platform over the next 5 years. And so we will select out some of the tests that we feel like are more likely to be used only in a physician-patient relationship or embedded in a treatment plan that's already exists. And we'll seek reimbursement for those. But the others are likely to be paid for in other ways, either by individuals themselves, or if you think about some of these tests. So, for example, we have a test in development that will tell us if a diabetic has a risk for a secondary complication in the next 3 years. And if you think about if you're a health system that's managing that risk, or if you're an insurer, the ability to take all 100,000 of your diabetics, run this blood test on them, and then know which 4,000 of those 100,000 are most at risk. This is information you don't have now, by the way, or the information that you have is very crude. Then we will likely also to be paid to use these tests in more sort of a population or care management way. And there's a lot of things that are paid for in this context now, like software, care managers, bolt on to EHRs and so forth, that are paid for, that aren't necessarily reimbursable. They're paid out of CapEx or OpEx. And so it'll be a combination of these things over time.
This is not only being used as a diagnostic tool, but it's also being used as a research tool. You mentioned work you're doing around NASH, which is fatty liver disease.
Yes.
How does this test compare to, say, a liver biopsy in terms of process and cost and outcomes?
Well, first, just to mention about the discovery component of our business. And so basically, what we've been doing for the last several years is delivering this protein information to pharma and academic investigators as a discovery tool. So, and that's fundamentally different from the diagnostic, emerging diagnostic business that I just talked about. Although there is some crossover, and NASH is a good example, there are some of these tests that actually pharma customers want to use in the context of clinical trials. They not only want the protein information so they can go back and look at that and decide if there's drug development insights to be gained from that, but they also are interested in using these tests. So NASH is a great example. One of the impediments to getting a trial done for NASH is convincing people that they should have a liver biopsy on the front end. And so if you had a tool that could, with great, in this case, it would be, you'd want it to be really sensitive. In other words, you'd want your false negative rate to be really low. So if you had a tool that had high sensitivity that you could enter patients into these trials with, and then actually monitor the progress of drugs, well, then that would be wonderful, and that's what we have. And so, obviously, the risk of a blood draw versus liver biopsies aren't high risk, but anytime you start sticking needles into the human body, there are some risks involved for sure. So that risk is eliminated compared to a blood draw. The sensitivity is quite high. There will only be a few individuals out of 100 we wouldn't be able to recognize that they had NASH. So the cost is dramatically lower. The safety is higher. That just sort of goes back to this concept of the ability to use this sort of data to replace, in some cases, invasive or expensive technologies to get to an answer.
I understand you're also using the platform for work around COVID-19. How are you using the test there, and have any interesting findings come from it?
Well, it's too early to know if we're going to have interesting findings, but we certainly hope so. And again, one of the reasons we have so many pharma companies that work with us, like Novartis and Amgen and others, is that you do get interesting findings when you're able to measure thousands of proteins at a time rather than hundreds. And so the first context that we're being used in here is we are working with pharma and academic partners. We are running their COVID samples for a number of them, and giving them the information back for their own use for disease characterization, drug development or vaccine development. So that's the thing we normally do in our discovery business, but we have shifted a component of our business. We have a special lab space, we have PPE and so forth. And we were prepared to do this because we've done it previously with other infectious diseases. We've got a segment, specifically dedicated to servicing pharma and academics that want to get insights, from the protein information. The second level of things that we're doing, we're talking to the government about, so that's great, but what else could we do since we have this ability with protein pattern recognition to potentially see risks that you couldn't see otherwise? And so we're also working to try to develop a test that will tell you if you're uninfected with SARS-CoV-2 or if you're just recently infected, what your risk of severe disease is. The problem with this disease, of course, is that, there's lots of problems with this disease, but one of them is that while a significant percentage of patients have known risk factors, for severe illness, there's a substantial minority that do not. And then that's the most frightening thing about COVID-19, right? Is that, with influenza, if you're not, elderly or and somehow at risk because of comorbid conditions, you just sort of you get your flu shot, and if you get the flu, it's not too bad. You're going about your life. The likelihood of someone with no underlying conditions dying of the flu is exceedingly low. It's not exceedingly low with COVID-19, and so how do you identify that risk? And there's a couple of reasons that would be important. One is that we could potentially get people back into the workforce more quickly without them having to have been vaccinated or have been infected and have at least temporary, as we think now, protection from reinfection. The second thing is who gets the vaccine first? I mean, this has been a big question in the media. Who do you vaccinate first? And so some things are obvious, right? Healthcare workers, people over the age of 80, people with immune compromise. But what about everybody else? Because we know that you or I may be at high risk. We just don't know it, even though we don't meet any of those criteria. So that's a great application too. And then lastly, even in people that are recently infected, who's at risk for severe disease and who's, if we can find a drug, and we haven't yet, but, I've actually got great faith that we will in the next several months here. If we can find a drug that taken early in the course of disease could reliably deflect severe disease, well, then who gets that drug and who doesn't, especially if it's in limited supply or expensive or has side effects. Lastly, just mentioning that because these little aptamers themselves can recognize proteins, we've got a little skunkworks project we've been working on to develop somomers that bind to the spike protein of the virus, which could have therapeutic or diagnostic import. It's going to take us longer to get to, but it's just worth mentioning that that's part of our work as well.
I think many of us have been anticipating the advent of precision medicine in a fuller way than we've seen it. What do you think your platform will do to move us toward that?
I think there's a couple of things. One is, at the highest level, sort of at the basic clinical level, the ability. So that example I gave earlier, the ability to look at a population of patients where currently it's really hard to discriminate risk. Again, with clinical data, with EHR data, we can get a certain way down that pathway, but for a lot of things, the output that we get is relatively crude around, what the real risk is and who's really at biologic risk. I mean, that we're making guesses on the basis of sort of statistics in that way, right? I mean, it's a population-based approach that uses you're probably at risk because other people look like they may be at risk like this. But this is different. This is actually drawing blood from your body and measuring your proteins and using that as our information source. And so the ability to, again, an example of this 100,000 diabetics, to tell a health system or a national health service that these 4,000 patients are the ones at highest risk, and you need to bear down on these. And the 96,000, not so important. They can go on about their business. But if you don't, develop, more intensive treatment for these 4,000, a lot of these people are going to have a problem. So that's a huge precision medicine advance to be able to be that precise. I think at the level of the individual, what we've seen in a couple of early studies is that this information has a different impact on them than your traditional, clinical advice, right? So when I was before I moved to Boulder, my one of my neighbors, a young woman, unfortunately got a genetic test back that said she had BRCA1. And so she didn't ignore that, right? I mean, she had a BRCA1 mutation, which meant she was at high risk for breast and ovarian cancer. And so she went and had prophylactic surgery in her 30s. And so because that was her body, her information, and somewhat immutable, right? I mean, it's like, you got a 90% risk. You need to act on this. The problem with it sitting in a doctor's office and being told, hey, Joe, you're a little overweight, you're a smoker, you got a little metabolic syndrome, your glucose is a little high, you're not quite diabetic yet. You really should make these changes because you could be at risk for, a heart attack. But what I can tell you from having been a cancer doctor, a cancer surgeon, is that, the human impulse is to think that, well, that might statistically be true, but I don't think it applies to me, right? And so this is one of the problems, getting people to act on information to take more responsibility for their health. In two small studies, neither of which have been published, so I don't want to quote them, but they will both be published. We actually just got some data back yesterday from a clinical collaborator, working with patients around primary cardiovascular risk tests. The percentage of individuals that actually change their behavior on the basis of these test results is really high. It's more like that, single gene mutation information from genomics. The problem is that very few things give you single gene information risk. And we'll be able to reach into all those diseases using proteomics to tell people, if you're told that you've got a 1 in 4 risk of a heart attack in the next 3 years and you thought you had no risk, and this is based on your biology, not on some, look at 20,000 other people and their clinical characteristics, which, by the way, were all indirect measures of proteins. The impact on precision health from in regards to the ability to get people to change their behaviors could be huge. The last thing that bears mentioning around precision health is just the development of new therapeutics. We know because of genome-wide sequencing that we're now being able to find all sorts of genomic abnormalities that could predict, risk for some diseases. The problem is that drugs target proteins. And we also know that, the first round of biologic treatments, mostly directed at cancer, a lot of those drugs didn't work very well. And we were all scratching our head because I was a clinician when, the first round of these things came out. And we're like, well, this is a genetic abnormality that seems to be somewhat common in these people. Why doesn't the drug work? Because it's targeting the protein that gene makes. And so it should work. And they don't work most of the time. They don't work most of the time because a lot of the therapeutic targets that are alluded to in the genome, it's not the protein from the gene that is altered. It's that altered gene and protein affect another gene that has another protein. That's just called a pleiotropic effect. And so, a large percentage of diseases, if you can't measure enough proteins, you can't find the targets for the therapeutics. It's like looking for a needle in a haystack. And so I'd say the most granular level impact of this will be the ability to develop new drugs and new treatments.
Roy Smythe, CEO of SomaLogic. Roy, thanks so much for your time today.
Yeah, thank you. Great questions, and I enjoyed the conversation.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send an email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.